ask, what are ventricles? You know, we have different ventricles in our own human body. During embryonic life, now there are three important vesicles that develop. These three important vesicles, you know, is what we term as the forebrain, the midbrain, and the hindbrain. So the forebrain now is what we call the prosencephalon, which is the first, you know, brain vesicle. I told you we have three brain vesicles, just like a balloon when the nervous system is going to form. So we have three swellings, one cranially, the other one in the middle, the other one behind. So the one forward is what we call the forebrain, and that is what we call the prosencephalon. And that one includes the telencephalon and diencephalon. In each of these divisions of the prosencephalon, that means the prosencephalon must have divided into two another vesicles. That means it also divides into two vesicles, thereby making how many vesicles now? 4. Because initially we said we have 3. Now the first one, the prosencephalon, divides into 2 vesicles. You get my point? That is telencephalon and diencephalon. So in each of these vesicles there are spaces. So the first space in the telencephalon is what we call lateral ventricle. But remember, we have 2 ventricles, left and the right. Are you clear? So the left and the right ones, they have spaces, and so those spaces are supposed to be termed first and second spaces, or first and second ventricles, or left and the right ventricle. But because they are named in numbers, so you have first and the second. But which one are you going to take to be the first or the second? Is it the right or the left? That is why the anatomist decided not to call first and the second. Because if you say you are going to take the right to be the first, what argument are you going to place? If you say you are going to take the left to be the first, which argument are you going to take? And that is why they are called lateral ventricles. But in between the two cerebral hemispheres, there is a space, which is a single space between the two cerebral hemispheres. That space, since it is single, it is called third ventricle in the diencephalon, because diencephalon is, you know, a structure located in between the two cerebral hemispheres. You get my point? So you have thalamus and hypothalamus on one cerebral hemisphere. You have thalamus and hypothalamus in the other cerebral hemisphere. So since they come together in the midline, the space enclosed by these two structures on either side is a single one. And that is what is known as third ventricle. So that means we have first, second, then we have the third ventricle. So in the midbrain, you know, which is the mesencephalon, the second brain vesicle after the prosencephalon, that one has a tubular-like space. That tubular-like space is what we call the cerebral aqueduct, or cerebral aqueduct of Sylvius, which we will see in a short while. And then the last, you know, brain vesicle, that is from the hindbrain, is what we call rhombencephalon. Rhombencephalon is the third or the last, you know, cerebral, you know, vesicle or brain vesicle. And this one consists of medulla oblongata, pons, cerebellum. So the hindbrain gave rise to these 3 important structures. That means it gave rise to the part of the brain stem and the small brain, that is the cerebellum. The cavity of this hindbrain, or the rhombencephalon, is what we call the fourth ventricle. Are you clear? So now we have seen that all these brain vesicles that finally give rise to, you know, the nervous system, you know, consists of all these spaces. So now, the lateral ventricle, we can see that this is just a diagram of the entire spaces in the 3 brain vesicles. You get it? So if you now look at this one now, this C-shaped like structure, this one, is the lateral ventricle. Are you clear? So now I said the lateral ventricle is, you know, a C-shaped kind of structure, and if you look at it here, it is located within the cerebral hemisphere. As I told you before, it's a space inside each of the cerebral hemisphere. So it is made up of components. We have a body that is located within the substance of the cerebral hemisphere. This body has some extensions. There's an extension forward into the frontal lobe. So the body is located inside the parietal lobe, and the body lies immediately above or immediately below the corpus callosum. Are you clear? We've discussed about what is corpus callosum, a bundle of nerve fibers or axons joining the two cerebral hemispheres together. So the spaces, these lateral ventricles are located immediately below the corpus callosum. So now we have the body of this lateral ventricle, which is a space. It has an extension forward into the frontal lobe, and that extension is what we call anterior horn of the lateral ventricle. While I said that the body is located in the parietal lobe, and the body also extends backward into the occipital lobe. You get it? So this extension of the body of the lateral ventricle into the occipital lobe is what we call posterior horn of the lateral ventricle. Similarly, the lateral ventricle also extends into the temporal lobe. That extension comes forward and downward and forward into the temporal lobe. This extension of the lateral ventricle into the temporal lobe is what we call the inferior horn of the lateral ventricle. So that means we have how many horns? We have 3 horns. Anterior horn, which is extension of the lateral ventricle into the frontal lobe, posterior horn, extension into the occipital lobe, and then the inferior horn, extension of the ventricle into the temporal lobe. Are you clear? Good. Now we've seen that. So the space that is lying in between the two diencephalon on either side is the third ventricle. This one, if you look at it now, it looks like a donut. You get it, alkaiki in our old language, or algaragi in the Hausa language. So this space in between the thalamus and the hypothalamus, this is the third ventricle. So the third ventricle now connects with the lateral ventricle through a connection known as interventricular foramen of Monro. Are you clear? So this is what we call interventricular foramen of Monro. So on either side you have this connection. So you have one lateral ventricle on the left side connecting with the third ventricle, and then you have another connection of that lateral ventricle with the third ventricle, through that. So we have 2 interventricular foramina of Monro, one on the left lateral ventricle, the other one on the right lateral ventricle. Reason because once the CSF is being produced from the lateral ventricle, the CSF now gravitates into the third ventricle. From there now, the third ventricle connects with the fourth ventricle through this cerebral aqueduct of Sylvius. Are you clear? So this cerebral aqueduct of Sylvius now connects the third ventricle with the fourth ventricle, so that by the time CSF is being produced in the lateral ventricle, and after being produced, it passes through the interventricular foramen of Monro into the third ventricle, and then finally the third ventricle also adds up. It adds to the formation and quantity of the cerebrospinal fluid. So that means the third ventricle also produces CSF. So all the CSF that are being produced from the lateral and the third ventricles, they all pass through this narrow, pathway. That is the cerebral aqueduct of Sylvius before it finally gets into the fourth ventricle. So the fourth ventricle is diamond-shaped structure. Diamond-shaped in the sense that it has 4 sides. You have 2 sides above and 2 sides below. We'll see in a short while how it looks like. So now if you look at this is just the lateral view of the whole setup of the ventricles. So if you look at it here on this lateral view, it looks like a triangular kind of structure. It has one side here, it has another side here, and then it has another side here. It looks like a triangle by looking at it from the lateral view. So this is just one side, the superior side of the upper borders of the fourth ventricle. This is the superior side of one side, and there's another superior side of the other side. These are part of the borders that form the superior cerebellar peduncles, which we'll discuss later on when we discuss the brain stem. While immediately below here, these lower borders of the lateral fourth ventricle, you know, the lower borders of the fourth ventricle of one side, that is part of the inferior cerebellar peduncles of the brainstem, which I told you we'll also discuss. So you have one border here, and you have another border on the other side. So you have two inferior cerebellar peduncles going upward from below, connecting the medulla oblongata with the cerebellum. One coming from the left and the other one coming from the right. So they pass upward and laterally so that to go into the cerebellum. You get my point. Similarly, from above here, you have the midbrain. So you have midbrain connecting with the cerebellum. So fibers from the midbrain, they pass upward immediately to go into the midbrain, because these fibers from the cerebellum, they are just going out of the cerebellum. Those one that forms the lower border of the fourth ventricle, they get out of the medulla oblongata into the cerebellum. And those from the superior cerebellar peduncle, they pass out of the cerebellum into the midbrain, thereby connecting the cerebellum with the midbrain. So they pass upward immediately, so that they form superior borders, two superior borders like this, and then the inferior cerebellar peduncles, they form the two inferior borders like that. So that is why we said that the fourth ventricle is diamond shaped. Are you all clear? Are you clear? So we have foramina, you know, from the lateral side of each of the ventricle. This is one aperture here on one angle of the cerebellum, lateral angle of the fourth ventricle, and then you have another one on that other side. So these are what we call the foramina of Loschka. And the one that is behind from this point here, this angle, which is this one I showed you, that is what we call foramen of Magendie. They all transmit the CSF to the subarachnoid space around the brainstem and around the spinal cord. Fourth ventricle has what we call a floor and a roof, and then borders. So it has superior lateral borders and inferior lateral borders, those ones that I've drawn before. You get the superior lateral borders and inferior lateral borders. And then this is the floor of the fourth ventricle. And then the roof is formed by the cerebellum, because the fourth ventricle lies immediately behind the brainstem and anterior to the cerebellum. Are you clear? So we have all these relations to the... So actually, this is just trying to show you the flow of the fourth ventricle. So this is the flow of the fourth ventricle, and this flow of the fourth ventricle has important structures from the back of the midbrain. We have so many swellings in there, which you will definitely appreciate when you discuss the brain. I'm not going to discuss these structures here because you have not done the brainstem. When you do the brainstem, you will definitely know what are the structures that are lying directly on the floor of the fourth ventricle. So these are the borders I'm talking about. The superior lateral border here, and this one is another superior lateral border. This is the inferior lateral, this is another inferior lateral border. So this is just part of the boundaries of the inferior cerebellar peduncle. These are also forming part of the, superior cerebellar peduncle. So this area is being covered by the roof. This one is entirely covered by the roof, that is lying anterior to the cerebellum. And so the area, the triangle that covers this area here, if you can see that there is something that looks like a straight line here, stria medullaris, that's what we call it. It passes across, dividing the diamond shape into two triangles, one triangle above and one triangle below. Are you clear? So this upper triangle is being covered by what we call superior medullary velum, while this one below is being covered by another, thin membrane, what we call inferior medullary velum. So these are what forms the roof of the fourth ventricle. And you know, I told you that it is directly anterior to the cerebellum. So the cerebellum covers the superior and inferior medullary villi themselves. You know, so now the most important aspect of this lateral ventricle is the formation of this cerebrospinal fluid. So we have to know the CSF very well, because it is important in that there are so many clinical conditions that you can, you know, ascertain by mere analyzing the cerebrospinal fluid. So you can use the CSF for your biochemical analysis and for chemical pathology analysis. That is why CSF is important, because if the brain is being affected by infection, the CSF can also, you know, give you the signs of the infection or the different variety of microorganisms that are affecting the brain, something like the meningitis, you know. And so meningitis, the organism that affect the meninges, you know, so they can, some of the bacteria or the viruses, you know, you can find them inside the cerebrospinal fluid. And so now the cerebrospinal fluid circulation in the ventricles, as I told you, that the CSF, you know, is mainly formed, you know, by what we call choroid plexuses in the ventricles. These choroid plexuses are in both the lateral ventricle and the third ventricle, as well as also in the fourth ventricle. So these choroid plexuses, they are nothing but networks of blood vessels. And so they now form the cerebrospinal fluid from the third ventricle, lateral ventricle, and the fourth ventricle. So it may also be formed by the capillaries on the surfaces of the brain. So that means it's not only the choroid plexuses that form the CSF, but even the capillaries around the surface of the brain can also, you know, form, you know, the cerebrospinal fluid. The amount, you know, at any given time, if you want to quantify the amount of the CSF at any particular time, you know, it's around 150 mil. If you want to know the production of the CSF per hour, it's approximately 20 mils to 200 mils per hour. So in each hour, about 200 mils is being formed. But one will ask, what happens if 200 mils is being formed per hour, multiplied to 200 times 24? It will give you how much? About 2.5 liters or so. It's more than that, about 4.8 liters, something like that. But what it means is that majority, more than 90% of it is being absorbed. So it's only that 150 that is being remaining in the circulation. So as it is being formed, it circulates around and gets absorbed. So it is bathing the CNS, the central nervous system. That is the purpose. It cleanses, you know, the brain, the spinal cord, and other structures like that, so that at any given time, the surfaces of this nervous system is clean of any debts. You get it? So all the toxic waste now is being, so it's like the water is bathing the CNS as it is being produced. It is being absorbed and get excreted via the, you know, blood vessels. So now we've seen the CSF. So you can see this is the corpus callosum, as I was telling you before. This is the corpus callosum. And so this area, if you can remember before, I told you, this is where the lateral ventricle, you know, is located. And here is part of its extension into the frontal lobe. And this area is anterior horn here, and it's been covered by a septum. What I was discussing before, too, I told you that this septum is called septum pellucidum. So the septum pellucidum now is the one that covers, you know, the anterior horn of the lateral ventricle, you know. So this is the septum pellucidum covering this part of the lateral ventricle. So if you look at this side, this is your third ventricle. And this is your thalamus here, and this is your interthalamic adhesion. That commissural fibers connecting one thalamus of one side and thalamus of the other side. Anytime you see two identical structures being connected by bundles of fibers, that is what we call a commissure. You get my point? So that is part of the commissural fibers connecting the encephalon together. So we call it interthalamic adhesion, connecting the two thalami together. So we have our own third ventricle here. So you can see this is the area of the hypothalamic nuclei. Remember I told you the hypothalamus is not a single structure. It is made up of conglomerates of nuclei. And so it's located just below, the thalamus. And so here you can see this is the cerebral aqueduct of Sylvius. And so it connects with the fourth ventricle. This is the fourth ventricle here. You can see here now, this is just like the interventricular foramen of Monro here, bringing CSF from here down here. So it comes down here, and then from the third ventricle now it goes into this cerebral aqueduct of Sylvius before it finally passes into the fourth ventricle. So this is the superior medullary velum, this rope here, and this is the inferior medullary velum here, covering the lower part of the fourth ventricle. And so it comes down here, and this is part of the this is the posterior foramen of Magendie, this one that goes at the back. So on either side now you have that the foramen of Loschka on either side, as I told you before. So now we've seen the lateral view of the ventricles together. So now we've seen the CSF, how it is being formed and how it circulates, and we have discussed some of the importance of the CSF, and I've already also told you that the fluid passes into the subarachnoid space of the spinal cord, and the importance of this CSF is that if you want to make a diagnosis, when you think that a person has something related to the brain, like if you, during the epidemics of the meningitis, like in Africa here, there is a period, when we have an outbreak of meningitis in our environment. So especially it affects young children, under 5 mostly. So you will see that these infants, now if you can remember, we said that infants, they do have what you call fontanelles, anterior fontanelle and posterior fontanelles. They are the prominent ones. There are those on the sides, the lateral fontanelles, but the most important ones are the one, especially the one that is on the anterior side and the one on the posterior side. The one on the anterior side is bigger. It's also diamond shape. So when somebody has meningitis, the anterior fontanelle tends to become tensed. But in normal individuals, you see that it is flat. If somebody is dehydrated, if the baby is dehydrated, you see that the fontanelle is down. It sinks down. You get it. But if it is, there's an infection within the cranium, the fontanelle becomes tensed. You will see it's bulging fontanelle. Even if you use your finger to press it, the place looks like a very hard place, because of high increase in intracranial pressure. And so for you to ascertain the organism that is causing that raised intracranial pressure in the cranium, you lie the person on what you call left lateral position, and then you now take your CSF, you bend the hip, you flex the hip and the knee joint so that the knee faces toward the chin of that baby, so that you have the backbone bulge towards you. And then you now take a landmark from the midline, mid-sagittal plane. So you imagine a line passing through the two crests of the two ilia with your midline along the vertebral column when you flex the baby like that. So you now put your needle at the point of intersection. You imagine that line, and then you put it. That point corresponds to what you call fourth intervertebral space. Fourth lumbar is at the level of the L4 disc. So you now put your needle there, and then until it gives what we call a gip sound, then you are inside. So the fluid will be coming out, and then you take as much as you like, and then you put it inside a bottle, and then you take it to laboratory for analysis. By so doing now, you send it for CSF analysis, and then you do MCS, microscopy, culture, and sensitivity, and then the laboratory technicians or scientists will give you back the result and tell you what organism is responsible for causing the meningitis or whatever. And then the content of the CSF has a lot of content. It has a glucose level, protein level, the level of the minerals, calcium, phosphate, sodium, chloride, all these biochemical components are there. At times, if it is due to a hemorrhage, because at times when somebody has what you call subarachnoid hemorrhage, if you now tap the CSF, you'll see blood inside. So when you see blood in the CSF after drawing, you know that definitely there is bleeding in the subarachnoid space. So some people that have this, stroke as a result of maybe hypertension, somebody that has stroke in the clinic, you can do tapping of the CSF, and then you see whether the stroke is as a result of the stroke, as a result of the hemorrhage or not. Because the stroke mostly is being caused by either rupture of an artery or blockage of an artery. So that means it's either that the artery ruptures so that you have bleeding in the subarachnoid space also, or the artery is blocked. If the artery is blocked, now the area is devoid of blood supply, so one can have stroke. If the artery ruptures, there is no more blood supply still, so the person can still have stroke. You get my point? Especially on the motor, the arterial supply and the motor cortex that we discussed before. So now we've seen the benefit of the CSF, or the importance of the CSF, and I told you that the CSF is being absorbed into the blood, and it is absorbed through what we call arachnoid villi and arachnoid granulations. These arachnoid villi and arachnoid granulations, they are around the what we call dural venous sinuses. So they enter into the dural venous sinuses, and so the CSF is drained through the dural venous sinuses. We'll get to see what are these dural venous sinuses. It is also absorbed by certain veins that are related to the spinal nerves. We have some spinal nerves getting out of the spinal cord, and so there are veins around the spinal nerves. They are also participating in absorbing the CSF. So CSF is being absorbed within the cranium through the arachnoid villi and arachnoid granulation, and then also it is absorbed by veins related to the spinal cords. So you see, as I told you before, the CSF has a lot of functions. So as one of the functions that I told you, it forms something like a shock absorber. It makes the brain buoyant, so it absorbs any shock. The cerebrospinal fluid now is responsible for forming something like a water jacket around both the brain and the spinal cord, and so that it absorbs any shock that comes along with the brain during accident or whatever. Its function is similar to that of the lymph. As I told you now, it cleanses the CNS, just like the lymph also cleanses our own body for any infection. And it also regulates our intracranial pressure. You get my point, because it has its own normal pressure. So as I told you, it is applied anatomy. It's like it can be obtained by using lumbar puncture, which I described to you. And so you can use it to diagnose several disease conditions, like meningitis, hydrocephalus. Any cause of intracranial pressure can be deduced from this lumbar puncture to ascertain the CSF. These are all what I've told you before, concerning the volume of the CSF and the protein, the glucose content of it. These are the values of all this content of the CSF, the glucose level, protein level, and the chloride level. Sodium and other ions are also there. And it also contains some lymphocytes. The lymphocytes are part of our immune cells.